Good morning, everyone. Welcome to this first set of breakout section. We hope you all enjoyed your morning break and also the morning break sponsored by INSMED. My name is Jing Wen. I'm a lab director at Embry Genetics, and I will be your room host for track 4 section. Science and technology innovation. We are excited that this track will also be live streamed, and I would like to welcome all the remote attendees to the section. As a reminder, we have our remote app available, so be sure to download it and to use some of the exciting features such as live Q&A. So without further delay, I would like to introduce our first section, Understanding the Biology of Rare Diseases. Please join me welcome Ethan Perston, CEO of Pereira, and Steve Rodhams, Senior Director of Discovery Biology at Retrophin. Welcome. Let's get started.
Okay, thanks. My name is Steve Rodhams, and I'm Senior Director of Discovery Biology at Retrophin. I've been at Retrophin for about 4 years. We're a rare disease-focused discovery and development company, fully integrated company, meaning that we can essentially take drugs from early discovery all the way through clinical trials and out into commercialization. Virtual company, so we do all of our lab work through collaborations or CROs. But company has about 150 people in it now, and we're staffed at essentially every single function along the way. So we can take drugs from early development, take them through clinical trials, and then commercialize them. Ethan?
And I'm Ethan. I'm the founder and CEO of Prolara. We started about in 2014, actually. This is my 5th Global Genes. And we are focused on the drug discovery side of things, not so much the development and the clinical side, but really preclinical discovery, kickstarting drug discovery programs, usually for families that have just gotten a diagnosis and there really isn't a big research community focused on their disease yet. And we're based in South San Francisco.
Okay, so we're going to talk about the biology of rare disease, and the outline is pretty much as follows. We'll take you through the Diagnostic Odyssey. Really just touch on this because you heard a session across the way this morning on diagnosis and genome sequencing. We'll spend most of our time in the biological mechanisms of rare disease, so really what goes wrong in the disease, starting with the mutation and how we learn about that mutation and learn about the biology. And then that informs pretty much the therapeutic approaches and how we go after finding therapies for that particular disease.
So as you kind of got a glimpse from the last meeting, the Diagnostic Odyssey is something that probably everyone, maybe everyone in this room is going through or has gone through. I think maybe you know the general statistics. This could be a 7-year journey. This could involve a couple of dead ends, 2 or 3 misdiagnoses along the way. But essentially, if your disease is caused ultimately by a broken gene, then the real way to complete this odyssey is through some kind of sequencing test. And so this really is this process can be really symbolized. I mean, on one hand, emotionally, potentially as, is this ever going to end? Are you going to find an actual root cause of this disease? But really is a funneling process. So starting either with an entire genome, all 6 billion letters, or maybe just the exomes, just the portion, the couple of percent of the genome that's actually just coding for proteins, or maybe even specific tests or panels looking at known variants for groups of diseases. However you enter this funnel, at a certain point in time, hopefully you arrive at what could be a single gene mutation. Now, as we'll discuss, it can be way more complicated than that, but in many cases it can all of this angst and all of this journey can be boiled down to essentially one genetic defect, maybe even just one misspelled letter out of those 6 billion. And there's as Steve mentioned, there's thousands of rare diseases, and so essentially each disease can be its own complicated, long journey to understand at root cause how do you go from a mutation, misspelling in the DNA, all the way through molecular changes that happen, through changes in cellular physiology, changes in whole organism physiology, and each of those stories may end up being very unique to every particular community. So elucidating this is not easy. To figure out all the details for each specific gene, each specific pathway, this is an arduous process. Could take sometimes years of basic research, usually in academic labs, to just get a foothold on what's going wrong. How do I connect from a mutation to what's actually going wrong in terms of symptoms and disease? And so, we'll spend the rest of the talk really diving deep into how you draw this circuitous connection between a gene mutation and the disease itself.
And we should add that we'd like this to be interactive, so we don't want this to be a lecture. So ask questions, particularly if you don't understand something, so we can clear it up as we go. So definitely ask questions as we go. So kind of, first of all is what goes wrong based on the gene mutation. So you have that information now. You notice that, from your genome sequencing, you have a particular mutation, maybe in both alleles, if it's a recessive mutation, maybe in one allele, if it's a dominant mutation. But you have that information. What do scientists do with that? So we'll touch on that. So they can be loss of function mutations, gain of function mutations, or actually both. And so what we mean is, I think we'll go through each of these pretty much in a little more detail as we go through the slides. But loss of function is simply the mutation in that gene causes that protein to no longer function as it did or no longer be expressed. Gain of function can mean a lot of different things, but it's maybe simply a new activity or something new that happens because of that mutation. And sometimes diseases will actually manifest as both. By losing that function of the protein, it will have some other effects that could be considered kind of a gain of function. These mutations can be in protein coding regions of genes, which are called exons, or in regulatory regions, which are called introns. There's things called untranslated regions. There's regulatory elements. There's regulatory RNAs. There's lots of places where mutations can happen and cause varying types of effects. Maybe I don't want to insult intelligence, but I just want to make sure everybody understands. So as far as introns and exons, if we remember, our biology is DNA gets transcribed into RNA. In humans, usually that is a kind of a precursor type of RNA that contains introns and exons. Introns get spliced out to put the exons together, and the exons are the protein coding regions. So sometimes you can have mutations within the introns themselves that don't actually affect the protein coding region, but they affect the ability of that protein to be made because they're somewhat in regulatory types of regions. And there's untranslated regions. If we consider what a messenger RNA is with the exons, untranslated regions could be the region before the exons or the region after the exons, and those are also regulatory regions. And so you can have mutations in there. So again, they don't affect the protein coding region, but they affect the messenger RNA itself. Mutations can affect the function of the protein, so that protein can no longer do what it's supposed to do, or it can affect the levels of the protein or RNA. So if it's a mutation that affects RNA stability or splicing, then you may have a lower amount of RNA and therefore a lower amount of protein. Or they can just simply affect the function so that protein no longer works. And they can be anywhere from a single nucleotide change, and we'll talk more about that in a second, all the way out to large chromosomal deletions that delete some obligate types of genes. Any questions there before we kind of move on? Question maybe for organizers, are the slides available? We don't have printouts, I didn't print anything out, but are slides available online anywhere?
We'd be happy to make it available. I don't know if there's an official channel for that, but we'd be happy to.
So our contact information will be at the end, last slides, you can copy down an email address. Okay, so what goes wrong in my gene mutation? So we talked about gain of function, loss of function. On the left is kind of a typical example of loss of function mutation. So the purple circles are the protein that's made from the gene, represented by the box. And the plus basically means that is a wild type gene. So you can see the two wild type alleles give you normal levels of protein. And then you can have where one allele is mutated. And so you're not making any protein off of the one allele. And depending on what that is, this could be either essentially a silent heterozygous situation where the protein that you do make from the good allele is enough to do the job. And so this person would be a carrier, but would not show disease. And that's a recessive type of trait. And then if you have two, in that case, if you had mother and father, one with a mutant allele, one with a wild type allele, then you have basically a 25% chance of having two mutant alleles out of that. And in the recessive situation, then that would be disease state. On the right-hand side, or I should say well, maybe the right-hand side is maybe a better representation of dominant type or gain of function type of mutation. So same type of situation, but now you have a mutation that causes the protein to be made in some other way. It's still the same protein, it's just maybe a different mutation. But that protein is causing something else to happen. Maybe it's aggregating with some other proteins and things like that will cause some other function to happen in the cell. And that be considered a gain of function mutation. That could also be dominant or recessive. Anything else to add, Ethan, on that?
No, I think actually, so this is obviously an abstract case, so maybe we can look at an actual real-world example of this in the next slide. So, both of our companies actually work on this ultra-rare disease called NGLY1 deficiency. Maybe only several dozen patients have been identified so far on Earth. And this was a paper from 2014 that actually describes the first cohort of patients that were diagnosed. And the reason why I'm bringing this up is to illustrate what a loss of function, or that's sometimes a more technical term for a recessive disease. And so don't look initially at all of the jumble of information in the table, just look at those family trees at the very bottom here. And I think people here are all familiar with looking at these, right? So one of the features of a recessive or loss of function disease is that it's not present in either of the parents. So parents are carriers, and so usually are unwittingly and have no idea that they're carriers, especially if it's a really rare disease like NGLY1, which there may not have been a panel for in any, when genetic testing companies, offer panels of tests, this may not even be included because it's such a new disease to medicine. But one of the telltale signs that you're dealing with a recessive or loss of function disease is that it's not present in the parents, and it obviously is present in the offspring, but the ideal ratio would be you get 25% of your offspring. If two carriers have children, if they had 4 children, you would expect, if all probability worked well, you'd get one of those children affected. But not every family is going to have 4 kids, and so you'll have outliers, it looks like, where all the kids are affected, or all the kids are not affected. But the bottom line is you do not see the disease present in the parents. That's the hallmark of a recessive disease. And what does that really mean in terms of loss of function? Well, if you look at the last row, this mutations row of the table, probably is unintelligible to most people, but this is all, this mumbo jumbo here is really just a way to code what the problem is. So part of that information is what's missing in the DNA. So C189-1 del means that you have a deletion of just this one letter. Remember, you have 6 billion letters in your whole code. This is an example where one of the parents had a deletion of just one letter. And the other parent had a different mutation. But in effect, what it results in is a broken protein. And a lot of these mutations, you can see they're actually appearing over and over again, this R524X or R401X. What that means is that these are all examples of the gene being stopped abruptly short in its tracks. So maybe you only make 75% of the gene or 50% of the gene, and the bit that's left is not enough to cover the whole function because the part that's lost is obviously critical. So a lot of these are, we'll get into details of what that kind of mutation is called, but this is a very typical example of when you have a loss of function. It's essentially the gene is not, cannot be fully made. That happens a lot. And so you see a lot of these patients are carrying examples of that, something number X, which means the gene is cut short. So that's an example of a recessive disease, and most rare diseases follow this pattern of inheritance of being recessive. But there are diseases that are dominant, as Steve alluded to in the previous slide with the example of the abstract case. Here is a real-world example from Huntington's disease. So this is actually data I pulled from the original paper in 1993 that was able to convincingly demonstrate the pattern of inheritance, and the mode of inheritance, i.e. dominant versus recessive, but also what is the nature, the molecular nature of the defect. And what you're seeing here, again, the same family trees, and these are now multi-generational family trees. And obviously all the affecteds are filled in black. And what's immediately different between the dominant and the recessive case is that the parents are affected. One parent is affected. You could have two parents, in theory, I suppose, being affected as well, in which case 100% of their offspring would inherit a damaged allele. But in this case, you see one of the parents is affected. And again, depending on the number of children in each family, depending on the size of the generation, you would expect, based on Mendelian statistics, that you'd get half of the children affected and half unaffected. And it's not always true, but I boxed in red a generation where that actually is true. If you count up the squares and the circles and which ones are filled and which ones are not, there's exactly 50-50. And that happened in these two different families in two different generations. So this is, again, the hallmark of a dominant disease. And then at the molecular level, what's happening is that in the previous slide, I showed a case where the genes are being cut off abruptly. So it's like a sentence, you have a whole sentence, and imagine, you're trying to text the whole sentence and it gets cut off mid-text. This is a case where you're trying to send a text message and all of a sudden there's a whole bunch of other random texts get introduced because maybe you accidentally butt dialed or something. And so what's showing on the right there is an example of this gene, the gene that's responsible for Huntington's. What happens is that gene can accumulate a bunch of extra letters. The 4 letters of DNA, A, G, C, T. And so what happens in the case of Huntington's is that if you look at the distribution at the top, these are from normal, unaffected patients, and this is asking how many of these so-called CAG repeats are present in the Huntington gene of normal people. And you get a distribution. Some people have a little bit more repeats, a couple dozen, some have a little fewer, but they all come around, they all hover around an average around 20. If you look at the bottom, those are the Huntington's patients. And their repeat sizes have essentially doubled. The population, the distribution has shifted to the right. So it's telling you that the Huntington genes here has more material, more genetic information than it normally has, and that actually introduces a problem. That makes, in this case, that protein a lot stickier, and it sticks to other proteins. That's the gain of function, right? The loss of function is the protein loses its ability to do things, and no other protein can compensate. Gain of function is the protein now does something it normally doesn't do, and oftentimes that means messing with other proteins and messing with their function. So dominant diseases at a mechanistic level can be a lot more complicated, although by no means is it easy when you have a recessive disease. So we'll spend the rest of this time going into the rabbit hole of how things can get more and more complicated.
Yeah, maybe I could just add on on Huntington. One thing that's interesting is the CAG repeat size is somewhat diagnostic for disease onset. So the longer the repeats, the earlier disease comes on. And the CAG encodes for an amino acid called glutamine, and so this is an example of a polyglutamine or a polyQ type of disease. If you've heard of that, there's several different types of diseases that are caused by expanded glutamine repeats, not just Huntington's disease. In this particular case, this mutation is within the coding region of the Huntington protein. So, and it's kind of early on in the coding region, so it starts out making normal amino acids, then it starts putting in all these glutamines. And then it finishes and continues to make the protein, but that protein definitely has some, whether it's solubility problems or whatever, but it aggregates with a lot of other proteins in the cell and takes those proteins out of play. How to treat that or how to find a drug for that is actually a tricky thing because of what that protein is and what those repeats do. Question?
Yeah, I was just wondering if there's any sort of rule about whether autosomal dominant mutations tend to be more rare or less rare, or?
I don't know off the top of my head if that's true or not. Huntington's has what, 25,000 or so in the U.S. I don't remember the exact numbers. As a dominant, we just talked about NGLI1 as a recessive disease that has about 50. So I'm not sure if there's any steadfast rules or if it's skewed one way or the other or not.
Yeah, I think that, I don't know the number off the top of my head, but I do believe there are more recessive diseases than there are dominant. So dominants are rarer, but I don't think there's a correlation with, say, how large the population is or things like that.
Okay. Okay, so again, kind of what goes wrong, based on the gene mutation. We talked a little bit about where they could be, whether they're exons or introns or regulatory regions. We just gave the example of Huntington's, which is within an exon. It's within the protein coding region. Cystic fibrosis is another example of one that has a mutation within the coding region. On the left is the CFTR sequence where one of the more predominant mutations occurs, and that's the F508 delta mutation. This is essentially missing 3 nucleotides, so it actually shifts the coding frame, but it keeps it in frame so that you actually make a complete protein, but you've deleted 1 amino acid. So this phenylalanine deletion, the loss of that amino acid in that region of the protein causes a misfolding event in that protein. That protein's not stable and cannot fold correctly, and so it ends up getting degraded, and you don't have enough CFTR in the cell membrane where it needs to be to do its job. So that's an example of one in a mutation in an exon. There's cases where there's mutations in introns, and one example I put up here is Friedreich's ataxia, where there's a mutation. Again, it's an expanded repeat. This GAA repeat, I forget the numbers of them, but they're in the hundreds in the disease state. This is in an intron, and what it ends up doing is in some way affecting the RNA, whether it's stability or how that RNA functions or how the RNA can be translated into protein. But bottom line is you don't make enough frataxin protein in this disease. But what's interesting about that is if you could make frataxin protein, you should be able to cure the disease because the open reading frame is fine once it's expressed. So I think that's important, and we use those kind of clues as we try to figure out how to attack diseases like this. One of the popular ways to go after Friedreich's ataxia is to try to increase expression of that messenger RNA so that you get more protein made, because the protein's perfectly fine once it's made.
And as we'll show in the next slide, this is just showing you, okay, you can get mutations in the part. Oh, sorry, there's a question. Well, I was just actually, maybe you should ask because I was going to go off on a whole new tangent. But go ahead.
Well, it's true that we have an inherited RUNX1 mutation that we deal with our foundation, and there's a lot of phenotype variability. And I was wondering if that's more a function of the non-carrier parent and what they're contributing, or the type of mutation in itself, whether it's a deletion or.
Yes, I think you're bringing up a point that the technical term is the genotype-phenotype relationship, right? And so Steve kind of mentioned it with Huntington's, where sometimes that can be very straightforward. You've got more repeats, and so the disease is more severe. The time of onset is lower. Now, you would oftentimes, when you start to understand and catalog all the mutations and start to understand their effects, you'll start to see this phenotype-genotype relationship occur. And sometimes it's very intuitive like that. The more severe the mutation, the earlier the onset. But sometimes there's mismatches, right? Sometimes you might have evidence that a mutation is quote-unquote very severe when you look in the laboratory, under some measurement or some test, but then in the real life, in the real patients, that those individuals with the so-called more severe mutations maybe not present as severely as someone who's got a mutation that doesn't look as quote-unquote severe. So oftentimes that genotype-phenotype relationship gets very muddy because there can be other factors that come in. So there's what are called modifier genes. So yes, your disease may be caused by a mutation of one gene, but the severity of the symptoms, the timing of the onset, the irreversibility of the damage potentially, that all can vary from mutation to mutation, but also because of the influence of other genes, mutations in other genes. So unless you know your entire genome profile, both parents, you don't know if there are any modifiers lurking there that may mean that your child is less affected, even though another family might have the same exact mutations and be the same carrier status as you, but their child has a different trajectory. Figuring all that out is part of why can be such a complex journey for each disease, because really each mutation doesn't necessarily require its own therapy, but each mutation, compared to another mutation in the same gene, can cause a different kind of disease potentially, and so you have to be cognizant of that. So, like in the next slide, there actually, if you just think about anyone who's done any word processing, you can make all kinds of mutations, all kinds of typos can occur. Now, it's important to know where those typos are occurring. Are those typos in the kind of, the words, the actual text, or if you're thinking about like a play, are the mutations or the typos in the stage directions, right? Maybe so thinking about exons, right? That's like, these are the actual words of the text, right? And you have to get these right. And so you can have many kinds of typos, what we call missense mutations. So changing maybe one letter of a word, and that may make the word now completely different, make unpronounceable, make it meaningless. It all depends on the position of that missense mutation. Then there are these things called nonsense mutations. That was what I referred to a couple slides ago, where you saw like R524X, that designation. That means is that you've cut the protein short. That's what I was saying before. It says you were trying to send a text message with a 5-sentence or 10-word sentence, and the last five words got clipped off. That would be the example of a nonsense mutation. And then there are the examples of splicing mutations. So Steve was saying genes are kind of are quilted together based on fragments and bits of DNA. Some of those bits are called exons, and some of bits are called introns. The introns have to be kind of removed out, and the exons have to be stitched together. You can have mutations essentially in the stage directions that tell the cell, how do I organize this gene? How do I stitch all these exons together, and how do I remove all the introns? If you've got mutations in parts of the gene that instruct this splicing process, that can entail its own particular kind of disease course, and might actually be amenable to a wholly different set of therapies than a different mutation that may actually look very similar based on symptoms. So knowing the exact nature of the mutation matters, and what kind of therapy you would use, because some therapies would make no sense if the mutation is of a certain kind. And then, of course, just like, again, when you're using Word and typing a document or writing an email, you can make mistakes that are typos where, you hold down the key too long, and, those kinds of typos. And that's what we refer to as insertions and deletions. And that all matters because everything, the way your genes are organized, the actual, what we call the frame, right? The way that you read out the sequence, because you're reading out 3 bits at a time, but if you have a string of letters, which 3 do you start looking at? Do you start with 1, 2, 3? Do you start shifted at position number 2, 1, 2, 3? Like, how you read that frame determines what kind of protein, if at all, you would make. And so there are plenty of mutations that again, all these mutations are randomly occurring. Every human being that's born is born with 40 new mutations that weren't present in either parent. And most of those mutations are totally innocuous, because otherwise we would all be running around with a rare disease. Maybe we all are. But the idea is that all these mutations, the genome is constantly churning all the time and throwing off these mutations and producing these mutations. And some of these mutations happen, missense mutations or nonsense mutations, they may happen at different rates depending on where you are in the genome. So that's adding more to the complexity. But bottom line is there's a whole litany of typos that you can make, and these are just some of the more common examples of the mistakes that get made.
Okay, so kind of to expand on that a little bit is mutations, as we say, can affect the function or levels of protein or RNA. Within the same disease, you can have different types of mutations that do different things but all cause the same disease. So CF, again, cystic fibrosis, is one of those where we have a number of different types of mutations. I talked about the F508del, which is the number 2 on that gray bar up there, the type 2 mutation. Is what's called a trafficking mutation. So as I mentioned, it's a misfolded protein, does not reach the cell surface so that you don't have any CFTR activity in the cell. That is the most predominant mutation in CF, but there's a number of other mutations that do other things. We talked about the nonsense mutations, which are the no protein, the type 1s here, which have, for example, G542X. There are other types. So type 3 here is labeled here as no function. G551D is an example. That protein gets made normally, gets trafficked normally to the membrane, but it's what's called a gating mutation. So this protein is an ion channel which allows chloride ions to move in and out of the cell. But in that particular mutation, those chloride ions can't move in and out of the cell very well. So the channel does not gate properly. So knowing what type of mutation this is informs the scientists how to develop a drug against those mutations. And we're also kind of getting a little bit toward precision medicine here, right? So there's certain drugs that will work on certain mutations, but not others. So there's drugs that work on the trafficking right now to get that protein to the surface. And there's drugs that work on the gating. So there are 2 different medicines that work on CFTR. Yeah, question?
You said about the trafficking and to get it to the surface. Is it then biodegradable so that the misfolded protein isn't able to deposit in the
Yes. So in this particular case, in F508del, that misfolded protein gets turned over more rapidly instead of going to the surface. The drug, and I'm not so much advertising for Vertex on this one. I used to work at Vertex and I worked on this project. But the drug that's approved right now for this actually increases that trafficking. So it helps the folding of the molecule, helps that molecule be more stable so that it can make it through that pathway and get to the surface. Once it's at the surface, there is data to show that it lasts at the surface longer than some of the misfolding protein can make it to the surface. It's not enough. It gets recycled right away. It lasts longer at the surface. So the half-time at the membrane is longer because of that drug. So it actually kind of, the thinking is once it's folded correctly, it can actually survive longer in the membrane.
right now with the Amyloidosis Foundation?
Yeah, so in this case, yeah, so I don't need to say more about the drug, but I think really the point of the slide was that there is different drugs for different mutations in a disease like CF. Other questions? Yeah.
Just wondering, is it a moving target though? Because if you have secondary mutations, wouldn't that affect this picture, for example, that you just shown us, a person's individual situation?
Secondary mutations, you mean new mutations within, you have a 508 mutation and another one? Or
Within one's lifetime, you pick up X, Y, and Z different mutations.
yeah, I mean, things like that, I mean, yes, can happen. So I'm trying to think of if there's examples I know of. You know of anything, Ethan?
I mean, that's tough. I mean, everyone's accumulating mutations throughout their life. The challenge is that it's going to be random mutations throughout your body. So unless it's like a stem cell that gets it, which then is responsible for repopulating a lot of other cells, then whatever damage happens is going to be confined to like whatever cell accumulated that mutation, right? Much like kind of the cancer situation. Maybe what you're talking about is more along lines of you're born with, in the case of CFTR, if you've got 2 defective mutations, you have variability in your other genes, right? And some of those other genes affect trafficking. Some of those other genes affect, the function of many classes of proteins. So it could be that someone may be a better responder to a cystic fibrosis trafficking drug. It could be that there's a different response because they have a mutation in some other modifier gene that makes them more or less responsive to that particular therapy. But I think the idea here is that these 6 classes, they essentially abstract out to whatever protein we're talking about. So everyone here is probably imagining their gene and their disease and essentially transposing it onto this picture and asking how does this explain or fit with my disease? And essentially these 6 classes, are general in the sense that if you have a nonsense mutation, doesn't matter what the gene is, you won't make protein. And it doesn't matter what the function of that gene is. This happens to be a protein that is an ion channel that lives in the cell surface, but there's thousands of proteins, and each one has a different place it lives and has a different way that it functions. And so this is just an example of what are called membrane transporters. But again, those 6 categories: no protein, no trafficking, these all apply broadly. So cells are almost all proteins are generating in the endoplasmic reticulum, and then they have to be trafficked out, and they have to essentially be delivered to specific spots in the cell, not just the cell surface. That can go wrong as well. So just imagine your mutation, your protein, you can apply it in this matrix, and the bottom line is that you're going to have to find the right kind of therapy, because if you have 2 nonsense mutations, then you might have a different therapeutic you might have different therapeutic possibilities available to you than people who don't have those mutations, and vice versa. There may not be a silver bullet, is what I'm saying. Although that is obviously the goal, is to try to find a compound or a mechanism that would work across all of these different mutational classes.
Okay, does that answer your question? Okay. Was there another question? Do you have a question?
Mine was, when I see those chlorine ions, are those just free, not very unstable?
Well, I mean, it's salt. So it is an ion channel that chloride flows through, but you're going to have sodium and other counter ions around as well. But the channel is transport, or there's chloride ions that are flowing through the channel. Yeah, questions back here.
Our disease, primary biliary cholangitis, is a predisposition. So they've identified certain genes that give you the predisposition, but it doesn't mean you're going to get it. So how is it a mutation? Like how do you?
Yeah, so situations like that where there may not be a clear target, right? That this is the gene that's causing that disease, but there is, as you say, predispositions. So as you do, sequence analysis or population analysis, say, oh well, this population that has PBC tends to have some mutation in this other, but not all of them do, right? So it's more of a predisposition. It's not like everybody has that same mutation, which kind of muddies the picture a little bit as to how to go after the disease. So not all cases are as clear as a single mutation that really drives the disease.
Yeah, this would be a kind of typical monogenic case where it's one locus, one gene is responsible. There could be 1,000 different mutations in that gene, but it's pretty clear that this disease is traced back to this one gene. But then there are other cases where what we're calling the same disease can actually be triggered by mutations in different genes. Sometimes those genes are in the same pathway, it makes sense, but not always. And that can complicate things because you can get the same diagnostic label kind of from a clinician, but genetically speaking, it could be caused by actually different genes. And so if you didn't know that, then you can't obviously tailor a therapy if you don't actually know which genetic predisposition you're dealing with in that case.
And I think some of these diseases or sometimes syndromes, they're different genetic diseases, and they will parse out into probably at some point being described as different genetic diseases. I think one of the cases I use the example of is autism. So autism is a syndrome, autism spectrum disorder. It's a large syndrome. There are specific genetic causes now that are being found for certain subsets of autism, and so that will help researchers understand how to develop a drug for that subset. And again, that's kind of going more toward that precision medicine type of approach. Yeah, no question.
So what happens when you do have this mutation, but the disease doesn't pop up till whenever it feels like it, it doesn't pop up as a child. It might pop up in your middle years. So what's going on all this time? I mean, you can't get any medicine. You can't take free medicine because you have to wait. If you don't have a specific gene, how could you get on some preventive medicine?
Yeah, and those are tough situations. So disease onset and can be whether it's genetic and whether it's that disease onset is determined by the severity of the mutation or what that particular disease is. Sometimes you don't know, right, until the symptom comes on. I heard the question in the other session of, will there be a time when every newborn is completely sequenced and we know everything about all the diseases? Maybe. We're not there, we're not close to there yet, but there might be a time where you'll understand those predispositions and maybe you can do something about it earlier because now you know your genetic sequence and you know the predisposition even though you're not showing any symptom. Okay. Ethan, you want to take this one?
Yeah, so this, as I said before, there are many ways that you can break the genome, and some of the bigger ways you can do it is by sometimes what we refer to as chromosomal loss, and sometimes referred to as microdeletions even. And this happens usually, not always, but usually toward the ends or the tips of the chromosomes. They're more vulnerable to that kind of breaking. But this is an example of a disease, Phelan-McDermid syndrome or SHANK3 deficiency, where you lose a bit of the tip of chromosome 22, and it happens to contain the gene SHANK3. There's some other genes that may end up being lost as well. And so in these kinds of cases where you've got these deletion or chromosomal abnormalities, it can sometimes be difficult to understand what is the exact trigger, because if you're losing a chunk of DNA, it may not be that it was the loss of all of those genes in that chunk that caused the disease. It may only be attributed to the loss of one of the genes in that chunk. And then in this case, in SHANK3, you can also have an instance where you've got patients who present with a deletion, where they have a loss of function by virtue of losing the actual piece of chromosome. But also there are examples where you could also have Phelan-McDermid syndrome, but you actually have mutations, loss of function, damaging mutations in the SHANK3 itself. So the chromosome is still intact, but you've actually achieved the same effect, which is to lose the function of this one particular gene. So that's another layer of complexity here, is that you can have presenting with the same so-called disease, it could be caused genetically quite differently, because if you have a case where the whole chromosome is lost, in the cases where you know that it's just that one gene, that can help you with therapeutic approaches. But in other diseases, there could actually be 2 or 3 genes in that particular part of the chromosome that's lost that are contributing to the disease state, and that's that's not as simple a therapeutic mechanism to try to alleviate, because that may be because the way genes are located on the genome. Sometimes genes that do similar things are next to each other, but usually it's totally random. So if you lose a piece of chromosome, that can be particularly problematic if multiple genes in that stretch were required for function.
Okay, so as far as kind of understanding the biology of the disease, so we've talked a lot about the mutations, different types of mutations. So now what do we do with that information? How do we start to understand what's going on in the disease? We need to understand that biology so that we know how to attack it and develop drugs to it. So, once you have the sequence information in hand, obviously you can just start by asking, what do I know about that particular protein or RNA containing that mutation? There's literature out there, somebody studied it, okay, so what's it tell me? Just knowing, so for example, we talked about missense mutations, and that's a mutation where you just put in maybe a different amino acid, so you have one amino acid change in the protein. You typically can't really tell what that's going to do to that protein just by looking at that sequence. I know it's different, what's the problem? So we might be able to look and say, okay, this is an ion channel. It's a cell membrane protein that pumps ions through the cell membrane. Does that mutation make that channel more active or less active? Sometimes I can't tell just by looking at the mutation, so you really have to do the experimental biology to figure that out. One thing that scientists will do is start to develop models, and that will be cell models or whole organism models that will contain the mutation. And cell models can be either, you can either make them or you can acquire them from the families, the patients. And I should say that is a huge advantage to research. 5 minutes? Okay. Having access to patient cells is tremendously advantageous to research. I just want to put that out there. So, it takes into account much more than we can do by just trying to make that mutation in a standard laboratory cell line. So, we can knock down that expression, we can overexpress it, we can start to understand what different levels of protein do to the phenotypes in the cells, and we can do that at the cellular level or at the organism level. And then understanding how that, what those do is how the protein functions tells us how to attack it. Okay, so we're running out a little bit of time. I think we're pretty close to the end. Again, understanding that mutation tells us what to do. You can actually take multiple approaches for the same disease. So there's lots of different approaches, small molecule, enzyme therapy, gene therapy. We try those different approaches for the same diseases. And there's potential to discover, one drug even for many different types of diseases. That could, based on a phenotype that's similar across diseases, maybe this drug will work for multiple diseases. Types of approaches, small molecules, large molecules, that could be enzymes. So you've probably heard a lot about enzyme replacement therapies, gene deliveries. We won't go into detail of these because there's going to be sessions on each one of these. So I encourage you to go to those sessions of interest to you.
And then I guess obviously, you want to apply your specific case to this. So, the general rule of thumb should be that you want to do an all-comers approach. Unless there's a reason why one of these approaches just is disqualifying for your particular disease, you want to try to have something, in the fire for all of these. So in parallel, you want to be working on a small molecule approach. In parallel, you want to be working on a gene delivery approach, and so on. You don't want to pick a favorite when it comes to a therapy. You probably want to make bets across all of them, and then when one of them really looks like it's working better than other, then you can start to focus in. But at kind of the initial stage, you want to keep yourself open to all of these therapeutic avenues.
Okay, this is last slide, but I think, as far as how do we start discovering these drugs, there's a lot of different ways to do this. We can take target-based approaches. If we understand what target it is that we're trying to drug, we can look at that particular target in an isolated case. So we can express enzymes and look for inhibitors of enzymes. We can do computer-assisted drug discovery, where if we have a model of that protein, we can actually start docking compounds, molecules into the protein to say, do we think this would work? It's getting that sophisticated. And the other approach is phenotypic drug discovery, so just looking at the phenotypes that come from these mutations, whether those are in isolated cells, whether they're in organoids, which are a group of cells that are kind of organized as almost like a mini organ, like a mini liver or mini brain, or some examples, or even in whole animals. Phenotypic drug discovery in whole animals. And there's advantages to doing different approaches different ways. There's no one way to do this. I think they all play into it. Ethan, anything else you want to say?
Leave the rest of the time for questions.
Okay, yeah, we got a question.
What did you mean by make the models from the back? Meaning, in a dish?
Yeah, for example, fibroblast cells are one example. So you can give a skin plug, and those cells will have your mutation in them, and allow scientists to study the effects of that mutation on that cell in a dish. Those can also be converted into, if you've heard of induced pluripotent stem cells. So there's technology now to take your cells and induce them back into a stem cell-like state. And that allows scientists then to differentiate those into, say, a neuron or a liver cell or some other cell type, depending on where the disease is that you want to study. So having patient samples in that case is extremely valuable to be able to test the drugs on, do the biology and test the drugs on.
And then obviously you make patient avatars, right? Everyone knows that they have to make a mouse, right? That's the first. And people tend to think that mouse is synonymous with animal, but there are many other animals that you could be working with. So you can make a patient avatar, you can make a fly or a worm with your child's specific mutation, and essentially do a natural history study on those patient avatars to enable you to then do the discovery and all the other future work.
Yeah.
What would your suggestion be from patient samples for us to have or to take for ourselves or our family members? And how do we go about that, I guess?
We're getting into topics that I think are covered in a lot of these forums about, how to get in touch with researchers. Again, maybe I can advertise. I don't know if the Grossmans are here, but I know the Grossmans have worked to set up a stem cell essentially an iPS cell library or multiple samples of iPS cells that are now available to researchers. So foundations are doing those types of things. They're working with some of the companies that do this stuff, like Coriell is a cell bank. And so you'll work with Coriell to deposit those cell banks that are now available to researchers.
I mean, everyone in this room is building a patient registry. So on top of that, that means accounting for, biobanking, and so any kind of materials. So, skin biopsy, you get fibroblasts, those can be deposited at Coriell, then those could be distributed to any researcher anywhere. You want to maintain iPSC biobanks, then maybe the cost of that is a little higher. You have to work with the right kinds of labs, you have to have the right Q&A protocols in place. But then those reagents, those cell lines could then potentially also be made available to be distributed to your disease community. But then that requires whoever's making them to have all the MTAs and all the legal stuff in place so you can share. But as you're building your registry, you have to be building kind of your biobank at the same time.
Hi, can I just add that I'm from Coriell, so I'm the Director of Lab Operations.
Perfect.
The principal.
Please say your name and stand up and introduce yourself.
Hi, I'm Nahita, and I'm actually speaking in the next session. So I represent the world's largest repository of rare diseases, the Indigenous Human Genetic Cell Repository. Coriell is a non-profit, and we house a lot of the NIH's repositories. And so I'm very happy to talk with everyone. The next session in this room, actually, I'll tell you a little bit about how to do that. I'll be focusing more on the iPSCs, but of course, and hopefully my team will have a booth here next year as well. But we do work with a lot of different groups around the world to bring in samples, and I can teach you exactly what we do.
So don't go anywhere. Stay for the next talk.
Okay, I think time's up. Thank you, Steve and Ethan. That was a fantastic talk.
Thank you. Thank you.
A lot of questions, and then it's very educational, and I hope everyone enjoyed the talk as I do. So I hope everyone can jump into your mobile app and do a brief survey for this section, and we little bit over time, so you guys have several minutes to jump into the next section. Let's give last applause to our great speakers. Thank you.